what I'm going to be talking about today is predominantly looking at fungal communities, initially talking about how we can analyze them and then how we can use that metagenomic analysis to then potentially start thinking about looking at antifungal resistance at the resistome level, so global resistance within a community. So first, a little bit of background for those who aren't familiar with the microbiome on the microbiome itself. So estimates are ranging between an equal number, so 50-50, up to 10 times as many microbial cells in the human body as there are microbial cells. Generally speaking, at the moment, people are tending to fall down on there being between an equal number and twice as many microbial cells. As a rough rule of thumb, that's coming up at around about between 1 and 3% of the body mass being microbiome. We also know that from the Human Genome Project that there's only around 22,000 genes in the human genome, which, given that at the start of the Human Genome Project, the prediction was that there would be over 100,000 genes to fully describe human functionality, is somewhat of a shortfall. We do know that from various different analyses, though, that there are a potential 16 million genes in the human gut alone within the microbiome. And we've certainly found that there's a possibility of around 8 to 9 million genes just in the oral cavity. This is the potential amount, not the actual amount, but even so, if we scale that back down to the number of organisms that's in there, it's likely to be in the region of 3 to 4 million genes just in the human gut that are coming from microbial sources. And to date, we've discovered over 10,000 different species of microbe in the various different human microbiome that we're finding. So why is it important? Well, we know that it plays a role in health. It's protective against disease. It can prevent pathogenic bacterial species from colonizing. Has a number of functions in the homeostasis of an individual there. We know that if you raise germ-free animals, then they have a condition called failure to thrive, where they're not as healthy as their dirty litter mates in that sense. And so far, every single disease that we've looked at, we have found changes in composition of the microbiome. And examples here are everything from gut diseases like IBD and obesity and diabetes, through to autoimmune conditions like rheumatoid arthritis, and more recently finding associations with neurodegenerative diseases and even mental health issues like depression, schizophrenia, and so on and so forth. What we don't know for certain yet is where these associations are purely associative and where they are causative. And that's a lot of work is going on to that area at the moment, looking at potential mechanisms by which these changes could cause disease. That in itself has led to an increasing knowledge of how microbial genes might modulate fundamental human physiological processes. We now know that the microbiome is intimately involved in metabolism, energy acquisition, immune modulation, neurological development, and a number of other fundamental conditions and fundamental processes in human development and homeostasis. So as such, we can think about as being a superorganism or a holobiont, where we're more than just the human cells. We're also all the other microbial cells in there as well. We can start thinking about the microbiome as being an extra organ within the human body that provides its functionality. In terms of how we analyze it, there are two main different ways that are used currently. There's the older method, which is this amplicon-based process here, whereby we sequence a particular gene. Notably in bacteria, it's the 16S ribosomal RNA gene. In fungi, we tend to use ITS1 instead, which is these internal transcribes as spacer regions between different ribosomal regions. And we're then using these sequences, which have a number of constant and variable domains within them, to be able to then determine from a database taxonomy and relative abundance of species within a community. And this is usually effective down to the level of definitely genera, and in some cases down to the level of species, although in fungi it's only really usable down to the level of genus. The newer method that's coming through, which is providing us with a lot more information, is whole genome metagenomics. So in this particular route, rather than looking at one particular gene, we sequence the entire extract of microbial DNA that we have there. And we can then use that using a variety of different tools. We can then use that to determine what species are present there. And this means it's kingdom independent. So it'll pick up any kingdom that we look at, including the viruses, but notably bacteria and fungi at the same time. So we can look at relative abundance of all of those organisms together. It will give us everything down in terms of genus and species. And even we can start looking at taking it down to the level of strain variation as well. So it's giving us a lot more information. And as well as that basic taxonomy of information, it also starts giving us other information. We can now start to actually ignore the species completely and just look at the gene families themselves. So start looking to see what particular gene families are enriched, whether we're seeing particular metabolic pathways enriched or depleted, whether we're seeing particular virulence genes enriched or depleted, and what's happening to profile of things like antimicrobial resistance genes. So we're now starting to be able to pull out this functional information. So using this total microbiome DNA sequencing, we're able to pick up a huge amount of different information from the community there. So moving on from the microbiome itself, we know that the microbiome consists of several different kingdoms within there. So there's the bacteriome, there's the mycobiome, which is the fungi, there's the virome, which are the viruses, and within there we have the phagome, which is the bacteriophages. Notably, though, the vast majority of work in this area has been done looking at the bacteriome, so looking at just the prokaryotes that are within there. And whilst fungi are only thought to make up around 0.1% of the microbiome in terms of genes in the gut, we do know that they make up a high percentage of the biomass of this microbiome. So typically between 1 and 3% of the biomass of the microbiome is made up of fungi. And we also know that other sites, such as the skin or the ear canal and the oral cavity, have a diverse and abundant microbiome, which is potentially richer and contains more species than the gut does in that area. So there's a lot there to look at within the microbiome, and we're only really at the start of looking at what's actually present there. Just to give you an example here, so in 2016, there was around 1% of papers published on PubMed that had microbiome in the title and were specifically about the fungal microbial communities out of all the microbiome papers. Moving forward to 2020, that was still the same. It was still, microbiome was only at around 1%. And as early as this morning, I did a similar search, and once again, we find the same thing. So there's over 30,000 papers published in 2024 on the microbiome, and there were only around 400 papers that were published on the microbiome in that sense. So it's still lagging hugely behind prokaryotes in terms of how we're looking at it in that sense. So under normal circumstances, fungi are usually either not disease associated or actually beneficial when we're thinking about the microbiome. However, if we get depletion of the bacterium with antibiotics, or we get dietary changes and things like that, or other environmental changes happening, we can then see an increase in fungal species present and a change in those fungal species that are present. We also know that these fungal species can interact with bacterial species, causing diseases. And we know that the microbiome is a rich source of metabolites and other factors, so things like secondary metabolites and things like that, which can all be used by the microbiome and by the host. And the microbiome has also been associated with a variety of different diseases. There is some debate as to whether it's actually playing a causative role in those diseases, but generally speaking, evidence is steering towards the idea that yes, the microbiome is playing a role in causation of disease in some cases. We do have a variation in the microbiome by body site, much as we do for the microbiome itself. So for the bacterial species, we can see that a healthy microbiome will vary between the two different sites of the oral cavity and the gut. And so we'll also see variation between the oral cavity and the gut for the microbiome as well. And similarly, we'll see a variation of the skin too. There are a number of problems, though, when we're talking about looking at the microbiome, one of which is the relative abundance of fungi to bacteria within the wider microbiome, so that there are much lower numbers there, which means that there's a real problem in working out what's present there. We need to know what fungal species are present because we don't really know for certain what fungal species are actually present there and which ones are transiently coming through. Unlike bacteria, we do use fungal species quite a lot in food preparation, everything from using Saccharomyces species in baking bread or in beer, but also down to mushrooms and species like that we're actually consuming as foodstuffs themselves. There is currently a real lack of bioinformatic tools and general resources such as databases and things like that to use for doing these kind of analyses. So we really need to improve those. And similarly, we're not entirely sure what the different fungal interactions are with other microbes within the microbiome. And so that's something which we need to look at. And similarly, the fungal interactions with the host are much less well understood than we do with bacteria, although that's improving all the time. But as I mentioned, we have seen several pathogenic species which are associated as pathobionts within the human body. So these are microbes which are normally commensal, but given the right conditions can become pathogenic. And so we can see a number of these at different sites, and the different species that we find will depend on the different site that we're looking at. Notably, however, you can see here that we're only talking in terms of genus here rather than in terms of species, and that's because at the moment, using the ITS-based technique, we can only discriminate down to the level of genus and not down to the level of species. So talking about this method, typically we would, for eukaryotic DNA, we would either use 18S ribosomal RNA or ITS sequencing there. For microbiome, we use ITS sequencing because it gives us greater resolution and ability to discriminate what genuses are there, but it's still only able to give us phylogenetic diversity to the level of genus. And we've recently found that it gives us a bit of a problem when we're talking about relative abundance, because the number of ITS sequences varies between different species, from a single sequence up to multiples of even 20 per species, which makes working out relative abundance a non-trivial problem in the end. However, taking alternatives such as using shotgun metagenomics data for the microbiome, there isn't very much out there. I haven't been able to find less than 20 publications that mention it, never mind actually use it actively in the last 20 years. So it's really in its infancy in this area. So we've got this problem with this metagenomic analysis, that we don't have public data sets that have been extracted specifically for fungi. They tend to have been extracted more for bacteria, which means that we're going to underrepresent the amount of fungal DNA in there because of the differences in the fungal cell wall. We have a lower number of organisms within the community as a whole, which means that to fully explore the full breadth of sequences there, we need to take a much longer approach and sequence to a much greater depth than normal. And similarly, normalization is an issue. How do we normalize data with a low alignment? And then finally, binning is tricky. So this makes it an issue to create these metagenomic associated genomes, or metagenomic species pangenomes, these other ways of clustering and categorizing them. And it also makes it a lot harder to identify novel species or strains within the community itself as well. So we've recently undertaken a study which is currently under review with communications biology by a previous PhD student in our group, to have a look and see how can we actually improve the metagenomic workflow for microbiome analysis. And so we took a process here where we looked at different ways to perform the DNA extraction, and different ways to analyze the subsequent sequence read data, metagenomic sequence read data, with different bioinformatic pipelines, using two pipelines that were currently prepared, as well as a pipeline that we generated ourselves. So initially we generated a mock community of fungi, which consisted of 12 species. There were about 7 Candida species, a couple of cryptococcal species, a couple of Aspergillus species, and finally Saccharomyces cerevisiae in there. We then looked for a combined fungal prokaryotic DNA extraction method. So this was building on previous extraction methods that we would use for extracting DNA from bacteria in there, but adjusting the lysis step to release more of that fungal DNA into the environment. And the key to this step is to use a combination of bead sizes in the bead beating step. So as well as using the small beads that we typically use for bacteria, we also introduce a number of larger beads that will help to grind up the fungal cell wall. And what we found was, when we compared different extraction methods using both a chemical system that we use in the lab, a homegrown system, was originally approved for the Human Microbiome Project at Metagenopolis, and similarly combining that with a commercial column-based kit, so this is the Cargen PowerSoil Pro kit, we found that although the homegrown kits will give us a greater DNA concentration, the quality of DNA that we get out is not as good as it is from the commercial grade kits. Similarly, although the data isn't here, we find much greater variation between operators if we use the homegrown kit than if we use the commercial Cargen kit. So as a result, we now switch to using the Cargen kit for all our extractions, and with all our collaborators as well, because it then means that we get less variation in the quality and quantity of DNA between different sites that are doing the extraction. We look at the species coverage. This is using a standard ITS and PCR based approach. Then we can see that species coverage is increased if we use the PKS So this is the commercial kit extraction. So not only are we getting better quality DNA, that means that we're actually able to pull out more of the different species that we have within our mock community there. So we're getting a better coverage of what's in there. We're able to identify all the species that are within our mock community and all to a similar level. Notably, we're pulling out a few of these other species as well, which aren't actually in our mock community, but this is once again down to the quality of genomes which are published and available online for us to be able to pull into the database. And that these species are, that there's quite a large amount of crossover between the species. So if we actually look in terms of how these species cross over, then we can see that at the top here we've got all the species that are within our community. Then these are the species which are highly related to the species that are within our community, and then we have a number of species that it's detecting via other methods which are kind of parallel to our community within that. So now we then developed a reference genome catalog analysis route for analyzing what's in there. Now, this means we build up a catalog of fungal genomes that we're then using as a reference to then try and predict what species are present there, taking down a de novo route. In parallel, we tried this with two other techniques which are out there, two other pipelines that are out there, one being the human microbiome scan using a fungi light process, which has 66 fungal genomes in there. And then we have Metaflan 3, which contains 500 eukaryotic genomes in there, of which around about 100 at the time that we did the analysis were fungal genes. So already we have a much greater number of potential fungal genomes within our pipeline that we can sit there and compare against. And what we find there is that using the fungal catalog, we get a much greater diversity and much greater ability to define what's in our community than using either of the other two pipelines. We're also able to pull out specifically, now when we're looking at what the DNA is that's mapping now, we're finding that it's not mapping to a bovine genome map or to other animal maps or human maps. Over 40% of the reads are mapping to the fungal catalog. We do have a remaining around 60% of the genes which aren't mapping to the catalog, so these are unmapped reads. But once again, this is a combination of the quality of the DNA that's extracted, but also more likely to be the fact of the degree of quality of the reference genomes and the breadth of the reference genomes that we're able to include into this library there. And so it's then not able to fully pick out all of the eukaryotic genes within the community. We're confirming these findings at the genus level using ITS and 18S based approaches, and we can see here that there's not a huge amount of difference between the two different extraction methods, but in general, PKS is coming out as being towards the better end in general there, but it's picking up all the different genuses genera that we think we should be picking up. So it's all well and good to be able to do this on a mock community. We need to be able to do this on real life patient samples. So we then did an analysis of stool and saliva clinical samples from 3 different healthy individuals. Showing you here in the first 2 images, we're showing you the data for, in the first slide, data for oral samples, and we can see that we're getting extraction there with DNA concentration matching into and more than enough for us to be able to do the sort of metagenomic sequencing that we need to. We are, however, getting a very low alignment level, which matches into the idea that within the microbiome, we have only got this very low proportion of the microbiome, which is fungal, and that's in both the oral and the gut cavity that we're finding there. Similarly, this is the reason why when we're sitting there mapping our genes there, we're finding that bacteria are by far and away the most common microbe that's mapping in now. We're seeing fungi far less, although notably you can see potentially here that that there's more fungi in the oral cavity than there are in the gut in that sense. Also notably here, we have a big problem with human contamination in the oral cavity, which is coming from saliva, which is largely down to the epithelial cells, which are in the saliva as well. When we're looking at genus level relative abundance in the microbiome, we can see that doing 3 replicates for each sample from the oral cavity and the gut, we're seeing on each particular occasion, each particular sequence occasion, we're getting comparable results for each sample there. And we're also seeing really quite a wide breadth of different genera that we're picking up here when we're looking specifically at the fungi within here. So we're pretty certain that we've now got a tool and a workflow which is enabling us to extract DNA effectively from fungi as well as bacteria, and to then analyze that DNA using a fungal reference genome catalog, which we can then pull out what fungal species are present there. And that catalog could be used in combination with a bacterial catalog as well to be able to work out, relatively speaking, what's there for bacteria and fungi at the same time. So now we know what's there, we need to think about how we go on to analyze it. Now, those people that have done any metagenomics before now and looking at AMR are aware of the fact that there are pipelines out there to enable us to use metagenomic DNA to analyze the global antimicrobial resistance profile within a community. And this would happen either through one of these different pipelines, but the most popular one to use is the central one here, where we take our high throughput sequencing data and assembly, we compare it against an antimicrobial resistance gene database such as CARD or ResFinder or things like that, and we then get a relative abundance estimation for the particular antimicrobial resistance genes that are coming out from it. And from that, we can then develop images like this, the data like this that we generated for a paper in 2020, looking at differences between the oral cavity and the gut resistome. And you can find that you get a real profile to know exactly which particular classes of antibiotics are showing resistance within a community and what the degree of that resistance is. And we can do various different analyses like that. Similarly, we can also sit there and categorize these patients based on the profile of the resistance genes they've got, to then be able to pull out particular resistor types and demonstrate that we have a real significant difference where we can identify major differences between the resistome in the oral cavity and the resistome in the gut, to be able to pick out precisely where these things differ. And so we can then sit there and see a real difference in shape there. We have started to apply this to antifungal resistance. However, this is very much in its infancy. So the initial data, this is unpublished data that we've got now, that is just giving us a feel for the start of the problem here, where we've taken the same approach and used the MARDI database that Matt Fisher and Joe Rhodes developed, and been able to use that to then predict out what particular genes we're finding there. And unsurprisingly, we find in the healthy oral microbiome that there's a large degree of resistance genes to azoles in terms of erg11 resistance, and also to echinocandins, with both FKS2 but particularly FKS1 represented there. We also are able to sit there and show that if we look at data from, or sequence reads from, patients with liver disease versus healthy individuals, then we can see that there is an increase across the board in antifungal resistance genes that we're looking at in liver disease patients compared to healthy individuals, which is probably reflective of the fact that these patients are receiving antifungal drugs more commonly and undergoing fungal infections and the like much more commonly than the healthy individuals are. However, there are some key issues with this, not least of which is the fact that the database itself is quite small, and also given the fact that as Matt mentioned in his talk earlier on, the acquisition of antifungal resistance is significantly different in fungi compared to in bacteria. In bacteria, there's an awful lot more transfer of genes horizontally between species, so you get a lot more specific genes associated with antimicrobial resistance there. Whereas with antifungal resistance, it's more about mutations, gene duplications, things like that within the genome. So that makes it more problematic and harder to actually use these same pipelines to look at antifungal resistance. So we do have some specific problems in looking at antifungal resistance here. First of all is the lack of databases. There's currently only two. There's MARDI, and then much more recently there's a new one called ResFungi, which is using a similar system to the mustard database for antibacterial agents, of using a combination of proteomics and genomics to predict genes in that sense. We also have these differences in antifungal resistance acquisition. So horizontal gene transfer doesn't really play that prominent a role, and it's more likely gene duplication and spontaneous mutation that are an issue, which means we need to rethink how we're actually looking at antifungal resistance within these communities. And finally, generating new databases is tricky. We need far more genome sequence data, and also in parallel with that, we need evolution data of fungal strains. So like those sort of microevolution data to see how resistance is developing within fungal strains, to then be able to use that to inform the tools that we're then going to use to identify antifungal resistance within communities. So that'll just finish off with some acknowledgements here. It would be remiss of me not to mention in particular Nilu, who's the PhD student that did a lot of this work, and Vicky Carr, who did a lot of the original antibacterial resistant work that we did there. And then more recently, we've had Lucas, who's been working with a collaborator of mine, Sai, who's also here at King's College, and is the computational guru with a lot of this work, and the rest of his group as well have had inputs into that. And with that, I'm done.